So in the last video, we talked about all the digestion that occurs in the mouth. At the end of it, we had this glob of food called a bolus, and we were ready to swallow. We were ready to pass that into our pharynx, into our esophagus. And that first step of swallowing is oral swallowing, which is completely voluntary. You have control over whether you swallow the food in your mouth or not. And once you decide to swallow, you pass that ball of food into your pharynx. Now, there's three components of your pharynx. There's our nasopharynx, our oropharynx, and our laryngopharynx. And something you might note about the pharynx is it doesn't just connect with the digestive tract with the mouth, it also connects the respiratory tract with the nasal cavity and the mouth. But we don't want food to get into our lungs. So we have this little flap of skin called an epiglottis. And when we swallow, that epiglottis is pushed down so that it covers our trachea. It closes off that respiratory path, and it keeps us from choking. So that's pretty much it for the pharynx, which is a component of both the respiratory and digestive tracts. And now we're ready to talk about the esophagus. So once we swallow, food is passed into our esophagus where it meets its first barrier, the upper esophageal sphincter. And this sphincter is completely composed of skeletal muscle, but it's not under voluntary control. It's under reflex control. So opening of the sphincter is a reflex triggered by your oral swallowing. So once that food is passed through that upper esophageal sphincter, it enters the upper third of the esophagus, which is actually all composed of skeletal muscle. But again, none of that skeletal muscle is under voluntary control. You can't constrict your esophagus. That's all just reflex control. And then below that upper third of the esophagus, we have our middle third, which is a mix of skeletal and smooth muscle. And the bottom third of the esophagus is just smooth muscle. That's not under reflex or voluntary control. And at the bottom of our esophagus, we have our lower esophageal sphincter, or our cardiac sphincter. Now, the esophagus isn't designed to hold food. It doesn't store food, so that sphincter is not designed to keep food from passing into the stomach. That sphincter is designed to keep stomach acid from flowing up into the esophagus. And when that happens, that causes something really painful. It causes heartburn. So we want to keep that sphincter closed. So our bolus of food has passed through our cardiac sphincter, and it is now mushing around in our stomach. Now, even though our stomach seems like the primary site of digestion, it actually only has one really specific job, and that's to denature our proteins. So all the proteins that we eat are folded into these highly organized shapes. And I've drawn this sort of spiral here to indicate an alpha helix, but obviously they're going to be much more complicated, big, globular, fibrous structures. And as soon as those proteins are present in our stomach, they're going to activate these G cells. And G cells are kind of the boss of the rest of the cells in the stomach. There's also parietal cells, which are responsible for maintaining the acidity of the stomach, and chief cells, which are responsible for actually secreting the enzyme that denatures proteins. But the G cells are going to be what tell those cells that there's food in the stomach and it's time to start digesting. So the presence of food is going to activate our G cells, which secrete a hormone called gastrin. So our G cells are going to secrete gastrin into the blood. And that gastrin is going to go on to promote the activity of our parietal cells and our chief cells. Now, I mentioned a second ago that parietal cells are responsible for maintaining our low stomach pH, and they do that by sort of vomiting HCl into the stomach lumen. HCl, as most of you know, is a strong acid, and strong acids dissociate completely in water. So that HCl is never going to be HCl. It's going to be protons, and it's going to be chlorine ions. And those protons are going to lower pH. Remember, a low pH simply means that there's tons and tons of protons. And that's how our parietal cells are going to keep that stomach really, really acidic. But then we have our chief cells, right? And our chief cells, those are responsible for secreting a hormone called pepsinogen. I wrote pepsin there, but it's pepsinogen into the blood. Remember, hormones are always secreted into the blood. It's not going to dump pepsin right into the stomach. Sorry, I need to fix that. It's not pepsin, it's pepsinogen. So why am I saying pepsinogen? That's because pepsinogen becomes a hormone called pepsin, which is responsible for unfolding those proteins. But you don't want a hormone responsible for unfolding proteins to be flowing around in your blood, because then it would unfold the proteins in your blood. Anything that regulates the folding of proteins needs to be really, really, really tightly controlled. And so it's secreted in something called a zymogen. And a zymogen is just an inactive form of an enzyme. You can see the ogen at the end of pepsinogen. That tells you that pepsinogen isn't the active form. So just because this is really, really important to remember, we have our sleepy pepsinogen here. It's not doing anything. It's not unfolding proteins. But once it finally enters the stomach, once it finally enters those stomach epithelial cells and then enters the lumen of the stomach, it enters this really acidic environment. And that acidic environment created by the parietal cells is going to cleave our pepsinogen into the active form, pepsin. So they act sort of like an alarm clock for our sleepy pepsinogen. This high concentration of protons alters those intermolecular interactions, and it ends up cleaving pepsinogen and creating this active form pepsin, which is super duper good at unfolding the proteins in our food so that we can digest them, so that we can reabsorb those amino acids and use them to make the proteins in our body. Just note, the mixture of food and all these stomach contents isn't called a bolus anymore. It's called chyme. So we mentioned that G cells secrete gastrin, which promotes excretion of HCl from parietal cells and secretion of pepsinogen from chief cells. But I want to talk a little bit about how it specifically promotes HCl excretion from parietal cells. So here we have sort of an enlarged parietal cell, and on the bottom right there, we have a molecule of gastrin. The gastrin is going to bind to a 7-transmembrane protein, or G protein coupled receptor, which is going to activate a phosphorylation cascade that ends up phosphorylating an enzyme called carbonic anhydrase. So this is our little pink phosphate on our carbonic anhydrase here. And what carbonic anhydrase is going to do is it's actually going to take CO2 and water, and it's going to combine them. It's going to make bicarbonate. I will post a video on the bicarbonate buffer system shortly, so if you have any questions on exactly how CO2 is converted to bicarbonate, that should answer them. But now we have this buildup of bicarbonate inside of our parietal cells, and that can actually be used to funnel chlorine ions into the cell. So you remember that parietal cells excrete HCl, which means that they need hydrogen and chlorine ions to make that HCl. Hydrogen chlorine, HCl is a strong acid, so it's actually never going to be associated with that proton when it's in an aqueous environment. So then we can use the protons inside of the cell and the chlorine ions. They can be excreted into the stomach, and that is powered by an influx of potassium ions. So potassium ions are taken into the parietal cells, and protons and chlorine ions are pumped out. Now, this whole pathway can also be activated by histamine, which is why some people get kind of sick or have acid reflux when they have an allergic reaction. Histamine is going to do it a little different. It's going to use a PKA pathway while gastrin uses a PKC pathway. I'll post a video a little bit later on that details the differences between those pathways, but you don't need to worry about them right now. Alright, so that's pretty much it for the stomach. But before I leave you, there's one thing I forgot to talk about, and that is the pyloric sphincter. Hold on, let me pull it up. That is the second sphincter in the stomach. And the pyloric sphincter is essentially going to prevent excess food from being dumped into the duodenum. And that's regulated by actually the amount of food in the duodenum. So that's going to be, two factors are going to contribute to that. First is going to be the actual stretching of the tissue. So there's actually going to be nerves that connect the tissue in the duodenum and the tissue in the stomach. And when that duodenum is stretched, it's going to prevent more food from being dumped from the stomach into the duodenum. But there's also a hormone called cholecystokinin, which I'm going to talk about later when I get to the intestinal digestion. And that's the release of cholecystokinin is also going to prevent more chyme from being pumped from the stomach into the intestine. And I think that's it for today. We covered the pharynx, the esophagus, the stomach, and in our next video, we will be talking about intestinal digestion, which is where most of our nutrient breakdown occurs.